Welcome back to our class. It is M.Saidi, as usual. Today, you're going to talk about the prediction of paper 1. And then when you talk about prediction, things you expect to find in the paper. Use this video carefully. It might change your life in case of this subject. So these are the topics we expect in paper 1. The first one is chemistry of life, basic unit of life, cell division, plant and animal tissue, then plant organ, support and transport. Support and transport system in plants. Take note of this because many people are confusing it. Explain it. And then support system in animal tissue. Please take note also of this. So we are going to show you, I'm going to show you where exactly you can find these 33 marks, what to focus on, where to put more effort so that you score a distinction. Let's use maybe plus or minus 25 minutes. The first thing you need to talk about is chemistry of life. I'm going to use our book for distinction material. If you don't have it, just look for that link which has join just below this video. It shows join and then pick the books. Then after that, you'll be able to access those books. Next year, we are going to do grade 11. So we'll be live. We'll be having classes every single day. Don't forget to become a member so that you get exclusive information and help for your subjects. All right, this is the first topic which you need to understand. And based on our number of days or weeks we spend on this topic, we end up finding out that it has 33 marks. How do we, or where are we going to get these 33 marks in this topic? All right. This topic is a little bit easier and difficult if you don't look at it critically. Okay, this is the first topic. This one's water molecule. We will ask you this, but in most cases, these come in the objectives. The micro and macro elements. It comes in the objectives. It's very rare to bring an essay or a short question with the too many questions concerning about this. Yes. So spend time, but not that much time. Yes. All this. Let's go there. Here in this topic, in this subtopic, spend a lot of time where exactly I'm telling you to talk about. Guys, talk about these lipids. Lipids. Don't make a mistake. Carbohydrates, lipids, proteins. Those are the most important subtopics in this topic you must look at. Let's go into detail of each. Even these ones, talk about them, but don't make a mistake. You have to read this fully. Yes. Because we might bring 2 or 1. Yes. So when it comes to that, what are you supposed to focus on that? I've been telling you exactly what you're supposed to focus on when it comes to that. I've been going to all those schools, many schools, just try to help you out, see that you score a distinction. Yes. So when it comes to this topic, what are your objectives you're supposed to look at, and how are you supposed to obtain a distinction in that regard? So when it comes to this, don't forget that this prediction is involving all the provinces. It's not about one province or what. So whatever I'm talking about is going to help you to get a distinction. Let's go to the first topic. So carbohydrates, make sure that you know it. I say that make sure you know it fully. Yes. made up of elements, oxygen, carbon. Yeah, that's not that a big deal, but make sure that you know the types of carbohydrates. That is the monosaccharides, the disaccharides, and the polysaccharides with examples. Yes, make sure that you know this table with examples. Yes, monosaccharides, single sugar, examples, glucose and fructose. Then disaccharides, two sugars, maltose. I'm not teaching. If please find out. Then the functions of the the glucose. You have to know the functions of glucose. Where do you find them? Here. You find them here. The functions. Why are they used? Actually, all these ones are used for structure. Yes, they provide a structure. It's for structure purposes, like they build up the cell membrane. You understand? Yes. But so that is one. If they ask you about the proteins, the lipids, the carbohydrates, that's number one. Energy is another thing. Ne? Please don't forget about it. Ne? There is also another thing. Two answers, they go across all of them. Ne? Yes. So the functions of carbohydrates, know them. The classes of carbohydrates, know them. How do we test for these carbohydrates? Yes, but not that a big deal, because yes, not that a big deal. Yes, because this one's that much of the practical, but sometimes you bring a practical in a theory. Yeah, so but know those two. Then when it comes to lipids, the same thing. Make sure that you know the functions of the lipids. Yes, what are lipids made up of? The building blocks of lipids, which is glycerol and the fatty acids. Yes, please make sure that you know glycerol and three fatty acids makes what you call the lipid. Yes, what are some of the functions of the lipids? Know them. What is the difference between saturated fats and unsaturated fats? Yes, here is just saturated fats. They have single bond throughout. While unsaturated fats, they have double bond between carbon atoms. Yes, double bond somewhere. Yes, so please make sure that you know that. Then testing, not that's a big deal, but the uses of the lipids, make sure that you go when you know it. Yes, don't go without knowing it. You regret for nothing. Ne? You regret for what? For nothing. Yes. So please make sure that you know that. This is not that a big deal. Then we go to proteins. We go to proteins. Proteins are also important as carbohydrates and lipids are important too. So structure of a protein, cool, what is it made up of? Then the functions of proteins. Please don't make mistake without knowing at least not three of them. These functions, at least not three of them. Yes, you can use this book or you can use whatever book you have been using. Make sure that you know testing not that a big deal, but you can read it. Then, wow, enzymes. This is a big star. Put a star on it. Yes, make sure that you know the properties of enzymes. Make sure that you know the properties of enzymes, specifically action, temperature, then pH, all that, those factors. At least you know how do they operate. Key and lock hypothesis. Yes, if it doesn't come here, it will come there. Yes. The reason why I'm saying this, I'm not focusing only on one thing, slightly one thing, is because this is a prediction for the whole country. Those kids who are in grade 10 and grade 11, the things you cannot go without knowing. Yes. How do I get a distinction from that? Yes. So key and lock hypothesis, please make sure that you know it. Yes. How it operates, products, enzyme substrate complex. Yes. Make sure that you know that. Factors affecting enzymes, also make sure, as I said, that lipids, carbohydrates, and fats, lipids, carbohydrates, and proteins, make sure that you know them in full. Yes, because this belongs to proteins. So factors affecting the enzymes, we have talked about it. Please make sure you know how it operates. And how do these factors, in terms of the graph, that is the pH and the temperature. Yes, that's for the pH, and then that is the temperature. Please make sure that you know that. Enzymes, the daily life, yes, you can maybe know 2, 3, then that's cool. Then nucleic acid, this is not that a big deal. You can know it. I'm not saying that don't go when you don't know it, but it's cool when you know it. You might have 1, 2 questions in the objectives, yeah, so that at least you don't lose it out. But you put more effort there on those areas I'm talking about, so that your marks can be boosted to a specific level. Yes. Structure of DNA, RNA, no, not a big deal. This one is usually bring them. Sorry, this one we usually bring them in the objectives. We like it in the objectives on the table format, whereby we give you a story and then we extract. Yeah, so yeah, please, you can read them, but make sure that you focus more on the other side. So these marks, which will help you to get the 33 marks from what I've tried to explain. What about the basic unit of life, that is the cell? What are you supposed to focus when you come here? Here, you don't need to know the structure of the cell, plant and animal cell. Yes, in a simple structure. This is not that a big deal, this one. Yeah, know this, yeah, so that you know the difference between the plant cell and animal cell, know how to label this difference between plant cell and animal cell, you see. So please don't go. When it comes to the table, make sure that you tabulate nicely, and then you have those two differences, at least three differences amongst them. Yes. So make sure. Now we have three organelles you cannot miss. Cell membrane, yes, it's fine, but there are three organelles. These movements across the membranes, please also know them about the diffusion, active transport, and then osmosis. Yes, all these processes, please make sure that you know how they operate. Then diffusion, that's diffusion, osmosis. There are three organelles which you need to talk about. That's the first one is the nucleus. Know how the nucleus, how to label, draw and label. Yes, and then the function of the nucleus. Then you go, the second organelle is the mitochondrion. Please know how to label the mitochondrion. Yes, we say that it looks like a what? A hot dog. A hot dog, you know, a hot dog with the you call it with the mustard, you know, with the mustard. Yes, talked about it. Please know how to label, and then the structure. So when you see this line, you just know that that is a what? That is a mitochondrion. And what's its function? Please know it. Then we have to know. I'm not saying that you should not know these other ones. You must know them, but this is crucial. That is what? That is crucial. You talk about the chloroplast. Don't make a mistake to go to the paper without knowing a chloroplast, the structure of a chloroplast. Look at it, how it looks like in the micrograph. Yes. And then when you're drawing it in a 2D structure, it looks like that. It has these pockets. These pockets. So it's a major difference between the chloroplast and the mitochondrion. What is the function of the chloroplast? Yes, please know that also. So it's stated there. Yeah, once you know that, at least you have covered there. Now, this is a chance to score this 19 marks. You see that topic is too big, it's 19 marks. But this coming one is small, but also has 19 marks. So it means that we can maximize those 19 marks and then we don't lose it out. What are you supposed to focus more on when it comes to this? Here, the phases. Please know the phases. The structure of a chromosome, please know how to label the structure of the chromosome. The phases, when I bring the diagram, what does it show? These phases. The phases we usually like is this anaphase. We like to ask about anaphase. We like to ask about the interphase, the prophase. We like to ask about the prophase, and then also like to ask about the metaphase. These three phases, I don't know why, but we like to ask. Don't make a mistake without knowing them. I repeat, don't make a mistake without knowing them. Yes. Then yeah, that's it. You see, it's a simple thing, so you can get all the marks. Yes. Then here, when it comes to differences between plant cell and animal cell, in terms of meiosis, in terms of mitosis. Yes, what is the difference? And then meiosis and mitosis. So basically, that's not the big, but cancer, yes. No, cancer is very small, but know these two types of cancer, which one is which. And then how do we, and what is cancer, the definition of cancer, because here is few marks, sorry, it's a very small thing, né? Yes. And then how do we treat cancer? Yeah, that's it. Nothing else. You don't need to talk too much things here. Yes, that's it. So the topic is done, that one also. Then you go to the 28, the plant and animal tissue. When it comes to plant and animal tissue, in most cases, we bring more questions on animal tissue than plant tissue. Please make sure that you know, you go when you know these tissues, né? The squamous, the cuboidal, the columnar, the pseudopod, pseudostratified. The pseudo means false. I explain this. Go find the video on YouTube. Just type a title and then put at the end Thundereduc. You'll find the, or M.Saidi, you'll find the answers. They are explained. I explained these things sometime back. Yes. So at least you know those. And then you need to know to identify them. This is what? Skeletal muscles, smooth muscle, cardiac muscles. Yes. When they draw them, you have to know how to identify them. Connective tissues. So yes. Yes. Also, you have to know how to identify this. If they bring it, you have to know how to identify this. A neuron, we also like to bring about a neuron. I don't know why, but maybe because we bring this neuron again in grade 12 that much. So please also go when you know it. So basically, this one is not that a big deal also, but when it comes to plant cell, know the parenchyma, the collenchyma, and the sclerenchyma, the permanent and the meristematic tissues. Yes. So the functions of the xylem, the transportation of mineral salts, and then the function of the phloem, transportation of manufactured food, and the hormones. Yes. So the adaptations of a leaf. No, not here. Yes, we don't talk about adaptations of a leaf here. So basically, that's what you need to talk about when it comes to this. Maybe what we can talk about here is these are parenchyma, collenchyma, sclerenchymas. Yes. These are now systems. Yeah, wait. This, please know how to deal with that. Now this is it's 9 marks. So here you also need to talk about the structure. This structure, where is my structure? So okay, I think I'll talk about it when I come, but it refers to this. We like to ask about the stomata. Yes, stomata. So basically, the 28 marks, you can obtain it when it comes to that. Then what about the 9 marks when you talk about the plant organs? The plant organs, they are talking about this. The leaf, basically, we are talking about the leaf. Not that big deal. The leaf, the dichotomous, the dicotyledonous plants. So the dicotyledonous plants, then you can talk about this. They know how to label, or this. It's rare to bring this, but it's very common to bring adaptations of a leaf to perform its function. Yes, please make sure that you know it. Yes, make sure that you know the adaptations of this leaf. Yes, adaptation of a leaf. These are also adaptation. Epidermis absorbs water and mineral source. So what is it about? So you just talk about the characteristics, and then yes, you see this stomata. They like it. They like to bring it, and then like the one which is drawn, and then try to label it where gaseous section is taking place. So this is what I'm talking about. Yeah, this. So the opening and closing of the stomata. We like to bring this. Yes, stomata, we like it. Make sure that you know how to deal with that. This is also a stomata. Yes, structure of a leaf showing you how oxygen and carbon dioxide is entering and going out. Then, when it comes to I'm done with this. Support system and transport. So all that is support system and transport. So here we are also talking about, you need to know about the transpiration. Oh yeah, it's here. So guys, don't go without knowing the factors affecting transpiration and how do they operate. Here, the instrument which is used to measure the rate of transpiration, which is called barometer. We like to bring it in the objectives or the terminologies, the biological terms. Yes. And then how is water absorbed? When I was trying to teach this, I emphasized this, that age is very rare not to bring this. It's very rare. And then the transpiration, we like to bring the experiment concerning about the transpiration. Yes, we also like it. But also this one, very rare to not to bring about this. Is because kids, they don't know it. Yes. So how is this xylem? How does it allow water to be absorbed from the root until the xylem? How is this root hair is involved? How is this water being absorbed from the root through osmosis into the xylem? Yes. And how are the mineral salts basically through active transport? But they want you to know this. You have to know that. Make sure that you know it before you go. Function of phloem, xylem, we talked about it. But how is the xylem adapted to its function, and how is the phloem adapted to its function? Please know that. How is the phloem adapted to its function, and how is the xylem adapted to its function? Then the last one is the skeleton, the support system. Here they're saying support system, non-support system, and the transport system. Transport system is in paper 2. I'll come back for paper 2 and then I'll try to explain it better. But paper 1 looks a little bit difficult compared to paper 2. Paper 1 has some few topics which are a little bit easier, and then paper 2 has some big topics which are a little bit easier, but it has also this classification where kids don't like. Yes, just few topics, so definitely you have to read in detail. So I'll explain where to focus more. So when it comes to the skeleton, know the functions of the skeleton. You can't go to the paper without knowing the function of the skeleton. At least know 2 or 3. Yes, functions, don't make a mistake. The two types, the parts of the skeleton, that is the axial skeleton, the appendicular. Then we have 3 things which we like on the skeleton. That's the first one. Okay, labeling is cool, but the first one is the positions, this position of foramen magnum, and then the A spine, this the shape of the spine. Yes, this is shape of the spine to allow flexibility. The position of foramen magnum, this whole foramen magnum, just on this one, it shows if the organism can be able to walk on 2 limbs or on 4 limbs. Yes, so make sure that you know that. Appendicular, please know how we like to, yeah, we can bring this, because remember those muscles movement of the no longer bring them that much, but now the skeleton it's being replaced by this. So make sure that you know it very, 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 very well. Guys, I think that's it. The only thing which you need to talk about are the the experimental questions, and it's the same thing. Grade 11, grade 10, grade 12 is the same thing we ask over and over and over and over again. So guys, yeah, I'll just connect the experiment. There are so many videos I've created concerning about that, about the experimental questions or investigative questions. Please make sure that you go through them on Thundereduc. Yes. So guys, I think I'm going to stop here. Unless you have a question, drop it down so that I can help you out, and then you are able to pass and get a distinction in this regard. So see you when you see me again. Graph. Yes. The first thing is we always ask it is line graph, line graph, line graph, bar graph, and then pie chart. So now when you talk about pie chart, there is something which I need to talk about before I go to this, is percentage increase and percentage decrease. Guys, this, when they ask you percentage increase, is final minus initial over initial times 100. Then it's going to give you percentage increase or percentage decrease. Is it clear? Yes. If I started with the 20, I ended with 40, then I'm going to say 40 minus 20 divided by 20. Then I'm going to get 20. Is it 20? I hope so. I I hope so. Which is 20 out of 40. Ne? Yeah, basically it's gonna be 20 out of 40, which is 50%. So basically, is it 50%? 20, 20 to 40. So this, if I subtract, it's gonna be 20 out. It's 100%. So it's gonna be 100%. That's how we do. But if now you started with the 40, you go to 20 out of so you end with, so it's gonna be 20 minus 40 out of, because I started with 40, then automatically it's gonna be it's gonna be what? It's gonna be a negative. So that means it's a percentage decrease. So don't stress if you see a negative. So that's it. The number 2, line graph. Guys, make sure that when you are plotting, it is nicely plotted. It's nicely plotted. Line graph, this is Y, this is X. Make sure that you put units. Make sure you put units. Caption must have the X factor and then the Y factor. Yes. We don't have a line graph which that. We don't have a line graph of that kind. If you find it, it means that you swapped the axis. Yes. Make sure that when you do the scale is maybe 0, 2, 4, 6. If the table is showing you 1, 3, 5, 6, don't start 1, 3. No. The difference is not the same. So just say 0, 2, 4, 6, or 0, 5, 10. It's not a must that you're supposed to start with a 0. Yes. 10, 20, 30, 40, depending on the lowest figure. Yes. So make sure that the difference is the same. Then if it is a bar graph, make sure that these bars are the same size, and the spaces between the bars are the same. Or otherwise, you're going to lose marks. Then if it's a pie chart, make sure that if they give you percentages, maybe 10%, 20%, we don't plot with the percentages. You convert this into 10% out of 10 out of 100 times 360, so that you use the protractor and then you measure. I did dedicated the full video concerning about this graphical work and how do you score marks in exam. So I'm just doing the highlight so that you remember. When you go to the experimental questions, investigative questions, independent variable, dependent variable, in most cases, when they bring the graph, the box, the first one in most cases is gonna be independent, the next one is gonna be dependent. So it means that ID, independent, dependent, it goes on x-axis, and then this one goes on the y-axis. But read the question nicely, you will get it right. You will never get it wrong. Yes. I think, yeah. Then how do you increase the reliability and validity? Please also go, when you know that, don't just say increase the sample size. Which size is it? Which